Hey everybody, welcome. Dr. Gautam Prasad is joining us as the presenter for this workshop. He works at Google as he works in like, what is your department again? Sorry, I should have pulled it up. Or I could pull it up
I
right
work
now.
on one of the research teams at Google, and predominantly on computer vision algorithms, like understanding things in videos or images. It's a lot of
Awesome.
neural network research.
Yeah, so he's joining us today to teach us a little bit about, so throughout this mentorship program, we're going to have a couple workshops, and they're all going to be focused on some form of data science applications or real world problems. So today, our talk is going to be about data wrangling. So given a bunch of data that may be of potentially questionable quality, how do we actually coerce that data into something meaningful? So please go for it. You have the floor.
Okay, great. And just to give you some background, prior to Google, I was a professor doing medical imaging research. And since this course is computational biology and medicine, is that correct? I tried to add in a bunch of examples that are hopefully relevant to that. I'll start sharing some slides. Let's see.
Go for it. I'm excited.
Let's see. Okay, great. Are you able to see the slides?
Yep, perfect.
Okay, perfect. Okay, so as Nima said, data wrangling is sort of the way that we can take a large amount of data typically and shape it up into something that allows us to answer questions about the world, typically like scientific questions we're interested in answering. So I will motivate that a bit. Oftentimes, we'll want to use data. So for example, we'll want to understand like how many cells are in an image, or like what is the composition of video data that we have, or image data. And we can ask humans to go in, identify different objects, say what those objects are. That's called object detection. A more specialized version of that that's often used is called segmentation. And this is trying to find the very exact boundaries or very precise boundaries of different parts of objects. So on the right, this is an example of a human brain where different parts, anatomical regions of the brain have been labeled. And typically in the past, we could have asked a human to label different images, tell us what the objects are. For brain images, oftentimes we'll use a machine like an MRI scanner to get a 3D volumetric image of someone's brain. And it's represented as a bunch of 3D pixels called voxels. And we would train humans to be able to label and trace out these regions. They would go slice by slice in these images and put those boundaries in them. And they would need to be trained to be able to understand what those anatomical regions would be. It could take them days to do this. Different humans could do it slightly differently. Even for images like the ones on the left, a human only has a certain number of hours in the day that they're awake and able to label these. And the reason why both of these are important is maybe we want to answer a question about how do the regions in our brain change when we have a disease or as we get older? And the way that we can make conclusions about that is we use statistical models to give us confidence about answers. For example, saying that one anatomical region is larger when someone has a particular disease. And for these statistical models to work well, we want to feed in as much data as possible. And the reason why that's important is because we can have more confidence about the conclusions we make using these models. And we also can make them more powerful. And by powerful, there's a statistical meaning for that. And that's the ability for a statistical model to recognize a particular relationship or different changes between different groups. But because the data we need to feed in to answer these questions is so complex, it's oftentimes the case that we don't have enough money or we don't have enough time for humans to label all of these images to be able to make really strong conclusions. And a tool that we have available to us now is machine learning. And that can take a human phenomena or a human behavior and wrap it into an algorithm so that algorithm can automatically label a brain image on the right or a bunch of images on the left. And a big term that's very popular today is called deep learning. And deep learning refers to neural network models that are really large. We'll talk about them in another week. But basically what you need to know is this is some kind of technique. You can feed a bunch of human labeled data into it, and then it can automatically label images like this at a huge scale. It could label billions of images, and it can do that in a speed way faster and way more consistently than humans can do. But to be able to create these algorithms, the first step is to have high quality, typically labeled data. There are a few other approaches where you don't need to label it by hand, but most approaches are supervised, and they need high quality labeled data to be able to feed it into this algorithm, to be able to automatically understand where things are in an image, or like to segment a brain. And because we have so many tools to create like deep learning algorithms and to implement them and to scale them up, oftentimes for a researcher or a scientist, most of the effort is actually spent in data wrangling and the data processing pipeline to get that data high quality enough so that we can implement some kind of machine learning method to convert all of this data into something that can be fed into some kind of statistical method to answer whatever question that we're trying to study, whether that be like understanding how a disease affects our brain, or a treatment changes the structure of our brain or makes people feel different, or understanding huge amounts of video or image content to help us answer a question about human life or biology. And I'll go over 3 different aspects of data wrangling. The first will be understanding outliers, the second will be normalization, and the third will be a more specific type of normalization called registration that allows us to compare different types of data together. And for each of these, throughout the slides I'll share, I have pointers to R and Python tools so that you can play around with these techniques, and also a few pointers to publicly available academic data sets so you can like pair the method with the data to try this out and hopefully use it in your own research. And also feel free to ask any questions. I'm happy to stop throughout this lecture and discuss anything. So the first topic I'm going to talk about is outliers. And there's a huge amount of aspects on this topic. So I'm going to talk about something that's hopefully easier to understand. Is when we have a data set, you can think of outliers as very extreme examples. But we have to be really careful about how we address these examples. So one way we could do this is look at our data and say like, okay, there's a bunch of really extreme examples. Let's throw them away because they're going to mess things up. And in this example, this is actually a plot of the salaries that law school graduates get during their first year. The x-axis is a salary range starting from around 40,000 to 180,000. The y-axis is the rank of the law school. The top is the higher ranked ones, the bottom is the lower ranked ones. The orange color represents a job in the public sector, and the blue represents a job in the private sector. And we can see here that the majority of jobs that people get after they graduate law school make about 40 to 100,000. And there's this really small, tightly clustered set of salaries at around 180,000. And you can see it basically represents people who have graduated the top 20 schools. And so at first glance, you could say, oh, hey, this is a bunch of outliers. Most people don't fall into that 180,000 category. And so any type of conclusions we make from this, maybe we should throw that out because they're going to mess things up. And that can happen. So for example, if we want to give a statistical picture of how much you're probably going to make if you graduate law school in the US, we could use the mean. And the mean will often be very skewed depending on outliers. And it might be closer to 180,000. And you might make the conclusion like, okay, I'm going to make a lot more money than is actually likely. And one way to get around that is you could say like, okay, the number of people who make 180,000 is pretty low. So let's throw that out of the data set and then recompute the mean. But in reality, those people who are making 180,000 are just, they are real people, and it's just shows how diverse your data set. And instead of throwing out the data, you should actually switch out the statistical method that you're using so that it is able to deal with these outlier data sets. And so in this example, a really simple technique you can use is use the median instead of the mean, because that's not as sensitive to these outliers. And it would represent what the majority of people are getting in the US. But this is sort of an example of like why you don't want to always throw out data if it works.
So there's a question in the chat. Would you use k-means clustering to make these types of conclusions?
Yeah, that's actually, yeah, I was gonna, that's something I'm talking about later, but you could do that. So another way to look at this data is it's a bimodal distribution. And so there's two basic groups that you could have from this. And with k-means, you might not know how many clusters there are, but there are techniques that you can use to see how well k-means is fitting your data set. So if you use two means, you could come up with two different clusters and say like, instead simplify this data into two clusters and say there's two main groups. And then you can make conclusions about those two separate groups. And oftentimes this is actually pretty useful when we're looking at different diseases, because a lot of times the models will not be able to categorize, like, for example, like big categories of diseases, like there's a lot of psychiatric illnesses where the definition is very fuzzy. And so maybe we feed everything into like bipolar or depression, but in reality, they represent a lot of more specific diseases. And we can use a clustering algorithm to actually come up with subtypes of that data and then model each of those subtypes separately. And yeah, so that's a really good point, that sometimes the distribution of data is much more complicated than the standard model will be able to understand, and we need to do more sophisticated technique like clustering methods at k-means. But the main takeaway I was that I have for this slide is oftentimes it's better to change up the statistical method than throw out data. Because if you did throw out those outliers, you might make conclusions about this data set that were untrue, because you actually, a human went in and just selectively removed samples, and you actually didn't have a fair representation of what the spectrum of salaries were for lawyers. Next, I'll keep progressively going into outliers that are more likely to be thrown out from your data set. So this is an example from MNIST, which is a really common data set that people use to benchmark machine learning algorithms. And it represents a bunch of numbers that are hand-drawn in cursive. And I have a link here from Kaggle, which is a platform for data science competitions, where people put up a data set and people all over the world try to compete to come up with an algorithm to recognize that the objects in that data set, or for this example, the numbers here. And the technique that they showcase in one of the participant showcases here is that they actually create an image for all the examples in this data set, and they went by hand and they looked for any of the examples that didn't make sense or were incorrect. And they created these giant images with thousands of sixes in them to say like, oh, were any of these sixes messed up? And then this is a zoomed in version of it, where they highlight 3 different examples. And you could see that the bottom left one right here is a 6, but maybe it could be a 0. And this might be a good candidate of throwing it out because the algorithm might mistakenly think this is a 6, but maybe a 0 would look the same way. It's sort of ambiguous. Whereas the top left and the bottom right, they could be a 6. They're likely to be 6 if we're looking at all the numbers. But if we also had letters in here, maybe they could be an L as well. And so these are sort of edge cases where it's more likely that we should remove them. And one technique to do that, as someone asked before, is k-nearest neighbors. So what you could do before you run these into a more sophisticated like machine learning method is you could run your k-nearest neighbor algorithm on this data set, and then you create clusters. And what you can do is in each of these clusters, you have a distance between the center of the cluster and the outside of it. And so you can pick those points at the outside of the cluster, and then you could inspect them by hand. So instead of looking at thousands of images and trying to understand which are the outliers, you can constrain it to maybe you're only looking at 20 images and understanding, hey, are these messed up? Should I remove them to my data set, from my data set? Another common approach is SVM, which comes up, one class SVM, which comes up with like a hypersphere that allows you to filter them out. Support vector machines was a really common approach before deep learning took over. And it's another approach for classifying different groups. And both of these links are to, I think, an R library and a Python library to be able to play around with them. And this gives you another tool so that a human doesn't have to go through all of these examples. Because maybe your data set has like 1 billion images in it, and there's no way you're going to be able to filter it out. But at the same time, you don't want to feed your algorithm a bunch of ambiguous examples because it might be just really difficult. Like there might be no clear answer to what the red box on the bottom left actually is. Like maybe it's a 6, maybe it's a smiley face. We don't really know. But at the same time, you could feed it into your, depending on the approach you use, you could feed it into an algorithm, and that algorithm could have a confidence measure, and it could just not be confident about the answer. But that's like another avenue to handle outliers. But this is sort of the edge of an incorrect example where we don't really know what's happening there. And then I'll give you another example. So this is also data from a Kaggle competition. The point of this competition is they give you images of cars on the road, and you're supposed to come up with an algorithm to understand the orientation of these vehicles with one another. And so they give you, if you look at these, all of these cars have bounding boxes, and you can try to understand the 3D structure of this scene and relate all of these vehicles together. And that's the data that you feed into your algorithm. But this is a giant data set, and one of the participants noticed on the bottom row that some of these images didn't download together correctly, or they weren't transferred correctly. And this is a clear error in the data set. We actually wouldn't want to feed this into our algorithm because there's no vehicles here, and the algorithm is going to be completely messed up by this. In some cases, whatever technique you're using might just crash because it doesn't even have like the car locations to do anything with. And so there's a bunch of ways that you can try to address this situation. Of course, you could look at every single image individually, and this is a pretty easy example to get out of. But there could be millions of images, and spending the time going through millions of images may make it so that you can't even do the project anymore. So one way you could do this, one really simple way, is you could use density estimation, or you could create a histogram of the pixels. And a histogram is the counts of the different colors of pixels. And on the top image, since it's a very naturalistic scene, you should have a smooth distribution over a set of different pixel intensities. But on the bottom scene, since half of the pixels actually don't have values, either you'll have a very low count of colors, or you might just have gray with a huge number of counts. And that's a really quick way to understand that there's a small cluster of images in your data set that should be actually thrown out. And you could use k-means clustering to do this, or actually there's a toolkit in Python called scikit-learn that walks you through a bunch of techniques that try to extract outliers from a data set that basically go through your distribution of data and help you navigate which samples look really different from the others. And then you can go in and understand, should we throw these out? But this is an example where it's like very clear you don't want them part of your data set because they will mess things up. Another more subtle case is this example. This is a Flickr data set from NVIDIA on the top left. And then the bottom is a cartoon data set. So oftentimes we might try to get some large data set of human faces, but some of those faces might be cartoons. And this is actually, that could be a really hard problem to solve because sometimes cartoons can look pretty realistic and filtering through all those faces could be difficult. And so one approach we could do to identify faces that we don't want is use a landmarking algorithm. And this might not be as sophisticated as whatever tool you're using to understand faces or answer whatever scientific question you need. But what you could do is create landmarks of where the eyes are, the nose, or the mouth, and then look at a small distribution of human faces and cartoon faces. And then you can compare those distributions, and hopefully it's the case that most of the cartoon faces just have a different relative position on all of those landmarks. And that can be a quick way for you to filter out images that might not work. Another really important problem that comes up, especially with brain imaging, is if you look on the left, we can take the same person and put them in two different scanners. And because of the parameters in those brain scanning machines, the resulting images can have a completely different intensity distribution. So on the left, the reference and the target actually represent the same brain, and they just look very different because they come from two different scanners. And one way we can solve this is we can look at the histogram of intensities and then come up with some transformation that makes sure that we can match the distribution of intensities in both images, and then use that to transform any images we get from scanner 1 to scanner 2. And they're more sophisticated approaches. For a brain image, there's 3 different types of materials found in them. There's white matter, gray matter, and the cerebrospinal fluid. And so what we can do is we can go to each of those regions in the image and then come up with their own histogram of intensities. And instead of globally trying to match these histograms, we can actually try to match each of those subtypes of anatomical regions and then come up with a more precise transformation between the two. And so you can see the normalized target makes it so that when we compare 2 images from 2 different scanners, we're actually trying to understand some kind of like anatomical difference as opposed to an artifact of the processing that was used to create that image. Another really simple example is if we have 2 different data sets, one is in color and one is in black and white or in grayscale, we can just convert the color images to grayscale to allow us to train something with a lot more data. Another example that'll often happen is how we parse our data. So what can happen with base images is oftentimes the pre-processing step used to identify a face in a much larger or detect a face in a much larger image will have different boundaries for how they crop the face. So these are 2 different academic data sets, and you can see on the right, there's a lot more room spent on the boundary of the face. And this is a case where the best solution would be to go through the processing steps and identify when the cropping step occurred or the face detection algorithm occurred, and make sure it's the same technique for both of these images. And this might mean that they use a different technique on the left because it's a color image and another technique on the right because it's grayscale. And maybe that means on the left, we need to convert it to grayscale and then run some kind of cropping algorithm to make sure that they are comparable with one another. And a big thing that could happen is if we were comparing these two groups of bases, our statistical algorithm might tell us just that they're two different colors or that they're two different groups because of how they were cropped. And typically we want to know more about like the content of the faces or the content of our data at a different scale, not focusing on the processing steps that got us the data that we can feed into whatever statistical method we make for a conclusion. Another really common approach that people will use before they feed it into a machine learning algorithm or statistical algorithm is some kind of normalization technique to deal with scaling. So these are two academic data sets where they focus on where a human is looking or gaze. And so in each of these data sets, we have an image of a face and then a three-dimensional vector for where the eyes on that face are looking. And one common issue that can occur is the scale used to represent that 3D vector. So on the left, maybe we have a position that is normalized to unit length. And on the right, maybe we don't. Maybe at each dimension, it can be from 0 to 1,000. And so if we feed all of this into our algorithm, we could have two vectors that actually represent the same direction, but our algorithm treats them as completely different. And it's only because of scale. And this is just an artifact for how the person who created the data set designed how they represent these directions. And a common solution to this is to do some kind of normalization. Oftentimes, generic features will be fed into L2 normalization. And there's a variety of normalization techniques that will enable two different features to be comparable. And the goal in this example would be to make sure that all of our vectors are unit vectors so that all we care about is the direction and not the magnitude of that vector. The next thing I'll talk about is registration, and that's a form of normalization that allows us to compare different images that have different shapes. And so I'll motivate this by reminding you about how when we use a statistical method to answer some question about humans or biology, we want as many samples as possible. So one situation where this is really important is genome-wide association studies. And the goal of these studies is we want to understand how does our genetic code influence our bodies and our life. And the way you can think about this is our genetic code is a bunch of letters. And we want to understand how do our genes affect, say, our brain structure. In our brain structure, we can represent as a three-dimensional image with 100,000 pixels in it. And our goal is to understand the relationship between this group of 100,000 pixels in our brain with 100,000 letters in our genome. And this is a statistical problem. But one of the issues that comes up here is a problem called multiple comparison correction, actually. So when we compare so many different possibilities, like 100,000 pixels with 100,000 letters, there's a chance that just by randomness, a bunch of them will be statistically different. Maybe 5% of them will be statistically different. And so the way that statisticians solve that problem is they use multiple comparison correction. And that means that when we get some kind of statistical result from a method comparing these two different pieces of data, we have to have a much stricter standard for being able to say it's statistically significant. We can't use the same standard when we're comparing 10 things versus when we're comparing 1 million things together. But the consequence of that is that these statistical methods become less powerful. And what that means is they don't have the ability to understand different relationships. So for example, if we want to study how does our genetic code influence the size of different structures in our brain, because there are so many letters in our genome and pixels in our brain images, the ability to understand that relationship means that we need to have, instead of 30 images, we need to have 30,000 images. We need to have really large data sets. And that means we need to be able to do some way to acquire that many images and then be able to compare them together. And so a sub-problem for this is, how do we wrangle all of these diverse data sets from all over the world so that they can be comparable with one another, and we can feed them into this big giant statistical analysis to actually understand the relationships between our brain structure and our genetic code. And one of the tools that we have to do this is registration. And registration allows us to take images of very diverse people of all over the world with different brain sizes and different shapes and make them comparable to one another. And the first step in this process is having a template space or an atlas. And this template, this is an example of a common template space or atlas. And this is a link to one of the most widely used brain imaging toolkits that people use in research. And you can think of an atlas as a map, a labeled map of all of the different points in our brains. So if we look at a specific pixel or voxel in a brain image, in our atlas, we understand exactly what that means and where that is in relation to all of the anatomical parts of our brain. And what we want to do is we want to take a new image and then transform it into this atlas space, so that for that new image of a person somewhere, we know exactly where Atlas 0.5 is on that image. And we want to remove the variability. So I'll go into a few more visual details for what that means. So in these examples, the red boundary is, you can think of that as your atlas. And that's the goal. You want to transform a new image into that atlas space. So you can kind of label all of the parts of it. And there's different transformations you can use to take someone's brain and move it into the atlas space. So one way you can do it is you can rotate someone's brain image to try to align it with your atlas space. Or you can translate it by moving it left and right, depending on how that image was acquired. And that represents a rigid body transformation. Another tool that we have is applying scaling. So if a brain image is larger or smaller, we can scale it up or scale it down. We can also look at skewness, and this represents a more complicated type of transformation called affine. And then on the right is the most complex types of transformation called nonlinear. And it's able to take shape changes and transform them to match an atlas. And what's really important when we do research is we can compute statistics over that transformation to fit a brain into that atlas space. And the changes and the transformation that we use to kind of push other people's brains into this atlas space allows us to do statistics. So we can say that there's a specific group with certain types of genetic markers that have larger regions in a certain part of the brain. And that allows us to compare very diverse data sets. And there's a bunch of algorithms that you can play around with to try to fit a brain image to an atlas and label it. And this is sort of another type of brain imaging technique or normalization technique, where if you took these images and you did your statistics on them without normalization, it could be that there were big differences between subgroups just because when they scanned people, a subgroup was looking to the left, or the scanner resolution on the images was a lot larger. And this is another point of whenever we make statistical conclusions about a data set, to go really carefully in all of the pre-processing steps and understanding, is there any point in our analysis where our conclusions could be motivated by different processing problems or differences in the data set that aren't related to, say, like in this problem, our brain structure, but really just the software you're using or maybe different sources of the data that propagate into our data. And that, yeah, that's all I had. If anyone had any more questions or wanted to discuss it.